do. It's a very broad overview, but it hopefully gets you some insights into some of the things that are happening now in the area of bioinformatics, which is where I work. So bioinformatics is a new field of work within the study of biology. It takes advantage of computers, of information theory, which is how do we handle knowledge and facts. And what we're doing is bringing these tools to bear on how we do biological research these days. I will take questions during the talk if there's something that's, you know, immediate, and I'll also take questions at the end of the talk, okay? I grew up in the area of the development of this idea of bioinformatics. It's tied together with the development of the Human Genome Project, and it reflects how we're working with biological information these days, and I'll talk a little bit about how it came about. So it's the use of computers, it's an emerging new discipline, it's the fundamental tool for 20th century biology because we have so much data. It's all of these things. Well, how did I get here? Okay, I grew up in New England in the US, in the Northwestern Connecticut, and then the last 25 years or so in the state of Maine. Here I am. I always have been working outdoors, started out as an environmental educator. Here I am about to figure out how to pack this kayak to go off sea kayak camping for a week. Now, although I live in Maine, I work in Maine, I live now in Colorado, so I'm much more in the mountains, doing things outdoors all the time. And all of this tied me together with wanting to know about the world around me, biological systems. And thinking back, I think it started for me really when I was about 10 years old, and I myself identified a northern cardinal in my backyard. And cardinals were very rare in Connecticut then. Now they're found all the way up into Maine, quite common. But for me, it was a spectacular mental accomplishment that I had figured out something that I hadn't known about the world around me. And birds have been a very important part of my life since then. Here's where I do work, even though I live in Colorado. I work at the Jackson Laboratory in Bar Harbor, Maine. It started as a summer retreat for scientists from Philadelphia and New York City. They would take ships up and spend their summers in Maine because of the no air conditioning from the cities, and they could bring their experimental animals with them, dedicated to genetics on the island of Mount Desert Island, and next to Acadia National Park. Those hills in the back are part of Acadia National Park, and the top left is the Bay of Fundy in Maine, in part of the system feeding into the North Atlantic. Science has always been about how do we look at facts, how do we examine data and reflect what we learn. And so for 1,000 years or more, people have been collecting information, typically in books and libraries. They work with their brain and to put this information into what I call it's called the reasoned argument, and they go from data to theories about that data to help us understand the world around us. And so a primary hero in that field has been Charles Darwin, who sailed around the world and part of the naturalist on board a ship investigating the birds and the animals, the plants that he found, and put many years of thought, and he came up with the theory of evolution. Just to remember that the theory is our current best explanation. If someone can, now the theory of evolution has had many, many, many years of study and a lot of understandings of the nuances. Still, if anyone could come up with a better explanation of all the facts that we see in front of us, or could falsify some of the key components, we would have a new theory. So anyway, the theory of evolution is where we summarize and integrate and reflect on the bits of information and data we have in front of us. Well, I have been working in this area of information management for a long time, but managing biological information is nothing new. I was fortunate enough after my graduate schooling and some other jobs that I held that I came to the Smithsonian Institution in Washington, D.C., where I was forever grateful to be working in the bird division. And this is where I started my world of bioinformatics, because I was being sent to Central and South America to collect birds, to understand both the diversity and the ecology of these birds. We would collect a certain number of them. We would make bird skins, and those bird skins would go into the national collections, as you see represented in this fantastic photograph. And the same time, I was in the early days of people being able to get samples of tissue and extracting DNA from those tissues. And in order to manage that information, I started building little databases. These were little desktop databases. But I immediately found out that the tags that were going on the specimens, and I came out of a museum community in my graduate work, they tagged the geographical location, the time, the date, all this information about these birds it was a file cabinet system at that point for these birds. I was capturing the same information in my notebooks, building little databases, but those two informations were separated from each other. When many of you probably have had exposure to some big sequence databases, if I wanted to find out the sequence or any sequence about these birds, there was nothing in the National Sequence Repository, GenBank or EMBL or any of these others that could tell me what the species was that the sequence data came from. It would say shark. It would tell you nothing about where it came from, what ocean. And so I started developing these little database systems to capture this information. So from the Smithsonian, I got involved in the Human Genome Project, then in the Mouse Genome Project, which is what brought me to the Jackson Laboratory. And I really focused on information management and how we worked with these data. My graduate school work had been around speciation and what DNA could tell us about the relationship birds within a speciation and between different species, how they were interacting. So this all was the way my brain works. And so I found fertile ground for a career development in bioinformatics. But the problem here is there are so many facts. We're generating more and more data all the time. Trouble with facts is that there are so many of them. So this became a focus of my work, is how do we work with this data and how do we look for provenance? Where did the data come from? How do we visualize the data? How do we reconcile different descriptions? Some description will say 3 miles down Route 73, and another will give a geographic coordinate space. So how do we align all these data so that we can look at them simultaneously? This is some DNA sequence data. Most of you have heard about DNA sequences, ACGT, adenine, cytosine, guanine, and thymine. And it looks, wow, look at all those, but it's only 4 base pairs. We develop computer programs that look for patterns. Maybe we're looking for a pattern of 4 A's, TGAAA. We can do that. That's pretty straightforward. But as my work tried to integrate all these different data sources, I started working with text data. This is called free text data, a scientific article. All these words in these articles can be different ways of saying the same thing. They can say things differently in one article and then another article. What does this term material derived mean? When this convention of citing the source paper for that data. So I also got involved in that. So genome annotations, genome scale data sets are most useful, as I've been talking, when they are integrated with existing biological knowledge. And that's what bioinformatics helps us do. These are my 2 main projects. The first one that brought me from Maine, from working directly on the human genome to now working on the mouse genome projects, is the Mouse Genome Informatics Group, which is an effort of bringing together all the mouse data that was being used to study human biology and disease, and organizing it in the context of biomedical ontologies, and I'll tell you about what ontologies are shortly. In particular, in a comparative way, because we use the mouse to study human biology and disease. There are many things in a mammal system like the mouse, which has fast generations and has many similar organs that we can do. We can try new drugs. We can do a lot of things in order to help us understand human biology and make all our lives better. The result of this work with trying to integrate information from the sequence data and from the papers that were published about these data in the Mouse Genome Informatics system got me involved in what's called the Gene Ontology Consortium. And I will explain what that all is in a few minutes. So this allowed us to organize this information in a complicated network. Think of a yarn ball, a network of information that the computers could access. And now we could use the power of the computers to help us understand and lead to new discoveries with that data. Data integration challenge, two main challenges I'm addressed here. The data integration challenge, where we have all these databases, as I mentioned before with the Smithsonian work. Each organism might have its own science database. All the medical centers might have their own databases. How do we integrate this information? We have to know when we're talking about the same thing, how we put all this information together. So this is the data integration challenge. It continues today. Most major laboratories have their own way of describing their sequencing experiments. That data is different from the way people describe the sequencing experiments in another laboratory, even though they may each be sequencing the same things or looking at the DNA sequence of liver tissue. They're not using the same words. In the Mouse Genome Informatics system, we start with the mice on the left, and we're studying human characteristics, as you see the suite of pictures of people on the right. And the trick with the mice is that we breed them together so they're very closely related. Human beings, each individual person's genetics has some similarities, but many, many differences from each other. Whereas in mice, because we can breed brothers and sisters, we can end up with a very restricted, it's called inbred mice. And here's this one here is called the stargazer. This down here is an obese mice caused by some genetic alteration. So we can study these conditions in mice and use them to think about what's happening in human beings. And what you're seeing here then is that we're using DNA primarily and different ways of working with DNA to uncover what aspects of the phenotype or what we're seeing on the external shape of all these people is a genetic component and what might be environmental or have some other origin. I mentioned the problem with text. Well, here's just an example of that. Here we want to search on tyrosine kinase, which is an enzyme. It transfers a phosphate group, which is a chemical, from ATP to proteins. And it's important in a lot of different biological processes, and it's important in cancer. And there's a lot of efforts supporting cancers these days. There are about 90 different tyrosine proteins in the human genome. So 90 different places on the human genome, you'll find one of these tyrosine kinases, which we identify by its sequence, but each one has a little bit different sequence. And if you go into the PubMed, which is all the place where everyone publishes their scientific observations and their manuscripts, there are now over 260,000 papers. Now, I don't have time to read 261,000 papers to learn about what I might want to learn about tyrosine kinase. So what I do and what the people I work with do is we develop these information systems that can tag these papers, mark up the information, and integrate what we have with other data. We do it using the power of computers and the power of the ontologies. Here's an example of the kind of thing we have to deal with. On the left is the ENCODE project, Encyclopedia of DNA Elements, and there are groups working around the world trying to catalog, like an encyclopedia, catalog all the different entities, all the different things that we find in the DNA. There's 356 terms about cell types in this effort. There's also another big effort. Both of these efforts, the Mouse Genome Database wants to get information from so that we can point between them and navigate on the web. There's another big project called the Epigenomics Project. So it's cataloging things that happen to the DNA in an organism, different kinds of changes that occur. So they use 314 terms related to cell types. The projects here, if we want to search by cell type, it seems like you should be able to interrogate between these projects. However, when we look at them, only 3 of these sample names are exactly the same between these 2 resources. So this is where data wrangling would have to come in, but wouldn't it be better if everyone was using the same vocabulary and to track this kind of information? It continues, so if you want to drill down and say find all heart-related tissues, what you're seeing here on the left is ENCODE has a bunch of heart terms. There are probably other terms around heart tissues. On the right with the Epigenomics Project, they have terms like fetal heart, right atrium, right ventricle, and others. So again, it's almost impossible to move between these 2 bits of information and find out what they share in common. So we need a controlled vocabulary. We need each group to describe their tissues using the same terms. And so this is the second major challenge that I'm addressing today, is the context challenge. We want to place all our data in the context of existing knowledge, but just listing terms as cell types or organs, it's not enough. So here we have a bunch of terms. You could carefully define all these terms or related to cardiac phenomena, cardiac phenotypes. The heart is a type of organ. There's a lot of blood vessel development as these organs are developed. And the 2 of the processes involved in developing the blood system are called angiogenesis, or new blood vessels, and vasculogenesis, which has to do with another part of the development of the cardiac system. What we want to do is develop a hierarchy where we can start with we understand that a certain experiment is describing what happens in vasculogenesis. This is a part, it's one of many parts of a greater process called blood vessel development, all of which is part of a process called organ development. So what happens then is if we have a term that's tagged as being part of vasculogenesis, now we can search by the term blood vessel development and we come up with all that data. So this is a hierarchical system. It's a complex hierarchical system. And the important thing here to note is that while this was really important development of these very strict terminologies for the mouse data, it has this also very important for all genetic systems like the the fruit fly and the yeast and all the different systems that were having their genome sequenced and now wanted to share and compare their sequence data. We all needed this hierarchy of terminology terms that is an ontology. It's very similar, and I'm sure many of you have learned about taxonomy. So here we have a fox. It's a canid, and foxes are mammals, which are also part of the vertebrate groups. And the same thing with birds, but at the higher level, they're all chordates. So you think, and at the species level, there are many species of vulpes, of foxes, different kinds of foxes around the world. So this is what we call a simple hierarchy. On the ontologies, which are a much more complex hierarchical system, what we're doing here is we're making a representation of some aspect of known biology. What kinds of things exist? Can we name them? What are the relationships between these things? So here you see an eyeball, and this white membranous ring around it is called the sclera. So what we do is we organize the information in the computer. A sclera is part of, it's one piece, one part of the eye, and we know the eye is these are the relations, it is a sense organ, and we know it develops from an optic placode. So in the development of the gene ontology, we not only carefully define each and every term, we also define the relationship of that term to other terms. So here, then, we result with a gene ontology. It's a big universal effort, a collaborative effort to develop a structured vocabulary defining the functional characteristic of gene products. Now, there's a couple things here before I go further. The reason that this was an important endeavor was that now we are comparing all life forms. All life forms are being described using the same vocabulary. In this case, we're describing the insulin protein or a gene product. This gene encodes insulin, a peptide hormone, vital in regulation of metabolism. So we tag insulin as some experiment, and we always track the experiments this data comes from, results in positive regulation of the glucose cycle, and insulin is also involved in insulin receptor binding, which is a type of, these little things are the relationship type of protein complex binding. It also is found in the extracellular space. And so we end up incorporating our biological knowledge into this hierarchical network of information. This representation over to the right, this figure is of the insulin molecule. Then, so as I talked about insulin, we can also do it with phenotypes. Here's a series of frog eyes, I believe. And each of these is tagged as being an eyeball in the ontology. An eyeball is a cavitated organ, and it is an organ. So if we wanted to search on cavitated organ, we'd come up with a whole series of different things, not all of which were eyeballs. So now we have the gene ontology, which can be used to mark up and search and find information from all cellular forms, as I mentioned, and viruses. We work with viruses as well. Right now, in this gene ontology, there's almost 45,000 terms. These terms have been used to annotate information from almost 1.5 million gene products. I call them gene products because sometimes people are investigating what we call genes, and other times they have a very specific protein. So gene products. In the gene ontology database system, there's 7.7 annotations, and they represent computationally available information for over 4,500 life forms. Taxa are different species. That's a lot of information. And this is all done by a group that I'm part of called the Gene Ontology Consortium. It has brought in many, many model organisms and agricultural organisms and plants and yeast, all kinds of different groups. And what we've done here is we're seeking to create the framework to capture biological knowledge and enable discovery. So it's a huge effort, many, many databases. Over here you see mouse genome informatics, my core work with the mouse community, but in the context of all these other systems. So we've all come and we're all working together, building the shared ontologies, the shared understanding of what all these words mean. The problem we were having, which we are solving here, is that before this age of computers, and particularly before the internet, now I am so old that I remember doing science, there was no such thing as the internet. fact, there were no computers back in the day. Like I said, we had little cards that we put the information on. So all these groups are now working together. They had all separate, they're all working with cellular biology and genetics, but each little group, like the group working on fruit flies, had their own dialect. It's like if you want to talk with, if you grew up speaking Castilian Spanish in Spain and now you go to Mexico City, it's going to be very difficult for the people in Mexico City to understand you and for you to understand, because it's dialects. At a higher level, we might say it's all Spanish, but there's all these dialects, and that was what was happening in the area of biological information, and that's what the gene ontology was seeking to solve, So these ontologies are really key to data integration because they allow us to not only identify the same entities, the same things that we have in each of our knowledge bases or our databases or repositories, but they also provide us with a way to name these things with computable definitions, with a classification system and relationships between terms that we can exploit. And so then we can aggregate all this data and do computational analysis. So this is the gene ontology. What this means, Darren, is that we can go to any of these web-based systems, we can go to the, and we can search on some enzyme that we're interested in. Here, calcium transporting ATPase activity. Okay, that's something that happens in the cell. All these different groups, from the fly database at the top, to the nematode database here, to the Arabidopsis plant model organism database here, we've all been tagging up our proteins, all the proteins, and we have a whole suite of them that we've identified as being involved in calcium transporting ATP activity. And now we can query across this whole system in a very robust, structured, and powerful way. Just to bring us back to the mouse genome informatics, here's a recent summary. These are the three main areas of the gene ontology: the molecular function, the biological process, cellular component, where it's located, and you can see overall all the different annotations for each component. So in our mouse system, we now have curated about over 24,000 genes, and I don't want to argue about how many genes there are, because this is another word that has to be carefully defined. The genes that we are describing here are not only protein coding sequences in the genome, but also functional RNAs. All of those are genes. So, and then we have, over 400,000 annotations, close to 30,000 papers captured in this system. Gene ontology started the ball on this back in the 1990s with the early human genome projects and the necessity to organize our information. There are now many, many, many biomedical ontologies. Here we see the mouse phenotype ontology. So the genotype is associated with the phenotype, and we can connect them all by understanding the relations among all these ontologies. That drives our ability to do computational analysis. And just some of the ways that ontologies are used. In the beginning, we didn't have the power or the depth of information captured that allowed us to do heavy computation on them, but we use them as standard vocabularies to share information. That allowed us to group data and start to do analysis. And we are now at a point that we ask the computers, what are you seeing here that we're not seeing? So here, this is an example of the gene ontology as a terminology. This is 2007. Genome-wide atlas of expression in adult mouse brain. And what they've done is they've isolated different tissues, and they looked at what kinds of genes were being expressed in those tissues and grouped those representations. See, it says selected go representation. So this tissue has high gene expression of genes involved in, say, lipid catabolism and blood vessel development. So this is a way that, because of the structure of the data, we're now able to describe and group and work with data sets. We can also use the gene ontology for reasoning. So here's a case, the rule-based reasoner, relatively recent phenomena, well, maybe 5 10 years, I don't know, but it's gotten better and better. We can infer new relationships, in this case between cell types, based on participation in shared biological processes. So at the top of the purples, this is lymphocytes and its cell types, and then this is the involvement in receptor activity. So there's the reasoner has told us, based on all the data, there's tons of data associated with each one of these little marks on the screen, mucosal invariant T cells are capable of histocompatibility locus receptor activity. So this is a hypothesis generated by the data we have in the system and the relationships we've mentioned for these data, that now can be taken into the laboratory and tested. So increasingly, these kinds of analysis give us the basis of forming hypotheses that we take into the lab to test. So what I've been talking about here today with you is data integration, what we call data integration via biomedical ontologies, which now drive computational approaches to discovery in biological and biomedical sciences. This is a huge advance from 25 years ago, or a little bit more than that now, when we initially were just getting to be able to sequence a human genome, to capture all the parts of the genome and being able to describe the functional parts, the parts that we didn't know anything about, and mark them up about what we knew about each one in each of these many, many genomes, and then put that data together and be able to deploy the computer to generate new hypotheses and to help us understand data and data interpretation. And the tools to be able to do this have just been developing very, very quickly, and so it's a real drive. In our recent times, I mean, it took barely a couple weeks for the new COVID-2 coronavirus for the genome to be sequenced, and for the tools available to compare that sequence to the genome sequences of other coronaviruses, to look at what was similar and what was different. The rapidity, the quickness with which we can do this kind of data analysis today compared to 30 years ago is just very amazing. And it's why bioinformatics, and it's all the ability to handle this data that is the work of bioinformatics that allows us to advance scientific understanding so quickly. Well, I just want to bring you back to where I started. For me, it all started with the birds and went into studying evolution and how we understand what different birds are. Here we're looking at the snowy owl, a bird that regularly winters now in Mount Desert Island in Maine. And I was working initially with classifications, understanding how we define different species, understanding how the species are related to each other in these phylogenetic trees, building all kinds of trees across all bird forms. Now I do the same kind of work, the same kind of information management, the same kind of bioinformatics, if you will, but working now with genetic and genome information. So what I want to leave you with, though, is the idea to just keep exploring. If you had told me when I was down in Panama that I was going to be working with genome sequences and understanding evolution in the context of DNA analysis and naming all these different proteins and all these different entities that we have in our genomes, it would have been science fiction. But here I am now, I'm still doing it. It's still exciting. It still fills my days. And I don't know where you'll find your excitement, but just keep exploring and keep getting on it. So thank you very much. And I'd be glad to take any questions. No questions?
There's one question, sorry about that, Judy. Question in the box is, although mice are very different from humans in that there's about a 3% gene difference between humans and mice, this would mean that a medicine that works for a mouse might not work for a human. So how is that thought, or how is that thought of throughout the experiments? So how's that thought of as they're doing the experiment?
Well, that's a very important point. I think in terms of organizing information, the bigger problem has been that the funding of scientific research has typically focused on organ systems. So take the heart. There's a lot of, and so a tremendous amount of research around the heart, and then we understand the information, and then we try a new drug, but nobody has connected the heart data with the fact that their same pathways are operating in the kidney. So we have a lot of concern and failures of developing drugs because they're not done in the holistic way of looking at the whole organism, but they're focused only on a particular organ. Now, that's within one evolutionary system, the human being. Now we want to compare human and mouse. How do we use mouse? Well, mouse have hearts. They have, and the hearts are very similar, but there are some differences between mouse and human. And there are, while the same pathways, if you consider most people have been exposed to the Krebs cycle, metabolic pathways, the same pathways are working, but what might happen in mouse is that there might be another enzyme. If you think of pathways of a stepwise progression to get a certain function accomplished, the pathway in human may be different. It may, at a certain step, there may be a duplicate protein available in mouse that would change the dynamics of how the drug worked in mouse or human. All this has to be taken into account. The more information you have about the mouse system and the human system, the more you know where the similarities are and where the differences are. But it's a very, very good question. But over the almost 100 years now that the mouse has been the premier mammalian model for genetics and genomics, all this data is incorporated into these systems. And one of the things that can go wrong is that when the developer of the hypothesis and the person who runs the experiments and the person who checks the efficacy of certain drugs doesn't take the time to fully understand the similarities and the power of the similarities, but also the differences. Many drugs are tested in mice first, or other organisms before they're tested in humans directly.
Thank you, Dr. Blake. We have another question linked to the last question. So will we basically figure this out once the clinical trials start in humans, or can we make projections in a holistic way?
Well, we can, and that's what's done with the mouse. A lot of work is done with the mouse, measuring blood work, and that's where we now increasingly, as we fully are able to get genome sequences very quickly, we're also able to insert or change specific nucleotides within the mouse and see the impact that has on the activity of a certain, like maybe a specific tyrosine kinase that seems to be dysregulated in a specific kind of cancer. We can now make the genetic changes in the mouse and see how that alters what's happening there. And so as I showed in the beginning with the libraries, we now have a tremendous amount of information that we bring to bear on our genetic investigations. I'm afraid I forgot it in there somewhere, the actual question again. Did I answer the question? Mouse has been an incredible organism to help us define all the functional units. There are some hundreds of different genes, or some genes that are duplicated in mouse and not duplicated in humans. There are some differences for sure, and that has to be taken into account as we do these experiments. But there are a tremendous amount of similarities that at least get us started and can eliminate some of the many choices that we could choose to investigate in the laboratory further.
Great. Thank you so much, Dr. Blake. Does anyone have any questions?